So I'm now recording. Okay, so starting with module 6, we need to know what the genome is. So a genome is essentially all of the DNA in the cell. This is going to include all of your genes. This is going to include all of the traits that are expressed in every organism. These are all within the genome. So of course, we have our two different types of cells. We have our prokaryotic and our eukaryotic cells. So prokaryotic cells, if we remember from the last unit, they do not contain a nucleus. So their DNA is in the form of a double-stranded loop of DNA, and there's only a single strand. So, and please bear with me, I have to use my finger to draw all of these. So if you need me to rewrite anything, just let me know. Okay, so in your prokaryotic cells, you will find a single loop of double-stranded DNA. And usually it's in the form of a circle. This is not really a great circle, but you get the point. Okay, and then moving forward into eukaryotic cells, they contain several double-stranded linear DNA molecules bound with proteins, and these are found inside the nucleus. So if we're to draw these out, we can imagine that these are a bunch of different pieces of double-stranded DNA. They are linear and they are within the nucleus of the eukaryotic cell. So that is how the genomes are in both prokaryotic and eukaryotic cells. So moving forward, we have chromosomes. So chromosomes, these are actually DNA, pieces of DNA wrapped around proteins. So I'll write that out. And in humans, we have 46 chromosomes in total, meaning we have 23 pairs of chromosomes. And these matched pairs of chromosomes, they are called homologous chromosomes. And essentially, these are the same length as their partner chromosomes, and they have specific nucleotide segments. And these specific nucleotide segments are referred to as genes. So genes are essentially going to be these nucleotide segments, and they're going to contribute to your traits. So they're going to be expressed in an organism, and they can affect how that organism looks or how that organism functions. So now going more specifically into traits. Traits are, like I mentioned before, a result of genes. So the genes can affect the traits that are expressed. And also, on top of being affected by genes, traits can also be controlled by environmental effects as well. So, for example, let's say, I would say skin color is a good example of this. We do have in our genes a specific amount of melanin. This is actually contributing to the skin color that we have, like the pigmentation that we have in our skin. But if you go out into the sun for a while, you know, people go get suntans and their pigmentation in their skin becomes a little bit darker. So this is a trait that can be controlled by environmental factors because your skin can have a deeper pigmentation due to the sun. Are there any questions so far? Okay, so we will move forward. So now we're going to go into cell reproduction. So there are a few different ways that cells reproduce. So in the case of prokaryotes, we're going to talk about binary fission. So with binary fission, I've listed the steps in order right here. So for the first step, we have the DNA replication of the single circular chromosome. So in here, we're gonna have the one single loop of DNA replicate itself, and that's why you see two different loops of DNA. And then the cell will elongate. It's not really clear with this picture, but let's imagine that the cell elongated. There we go. And the proteins are going to migrate to the center. Then, after that, the proteins will form a ring. So this is a ring being formed in the center. And this is going to create a septum to divide the cell. And after this, the cell will pinch. This is step 4. And it'll create two identical daughter cells. So essentially with binary fission, these bacteria cells are just going to keep making identical copies of themselves. And there's no growth happening in between. It's just replicating and replicating. With eukaryotes, it's a little bit different because these cells are going to grow, and then at one point, they're going to divide and make a new cell. And this process is called mitosis. So mitosis has essentially 5 steps, but I added on a 6th step. I would know these steps in order for the exam. And these steps include prophase, prometaphase, metaphase, anaphase, telophase, and finally, cytokinesis. And the reason why I separated cytokinesis is because this isn't technically part of mitosis if you look at the steps formally, but this is essentially just when the cell is being cleaved to form 2 cells. So the way that I remember the steps of mitosis is with this acronym, PPMAT. If you go to other resources, they may mention PMAT, but in this case, we are including prometaphase in between. Okay, so I'm going to go through each step of mitosis individually, but before we go into that, I just wanted to mention that in eukaryotes, there is that period of time when the cell is growing, and so this is referred to as interphase. So in interphase, the cell is growing, it's producing proteins, and it's not yet ready to divide. So there are 3 parts to interphase. There is the G1 phase, there's the S phase, and the G2 phase. So in the G1 phase and the G2 phase, these are when the cell is going to grow and proteins are going to synthesize. And in the S phase, we're going to have DNA replication. And in interphase, you would find the DNA loosely packaged in the nucleus. So this would represent your DNA, and it's inside the nucleus, and it's not condensed at all. It's loosely packaged. And so when the cell is finally ready to divide, it's finally going to go into the mitotic phase, where it's going to undergo mitosis. So the first step of mitosis, as we mentioned before, is prophase. So in prophase, this is when the chromosomes are going to condense. So remember, as I mentioned before, in interphase, the DNA is loose in the nucleus, and it's just in the nucleus, it's not condensed at all. In prophase, this is when it's starting to condense. So you would start to see, I'll put them in orange, you would start to see chromosomes forming. And of course, in the human cell, remember, we're going to have 46 chromosomes in total. And they are going to all condense and become visible because they are so condensed. Also, spindle fibers are going to emerge from centromeres. So centromeres inside your cell, which I'll represent by these black dots, they are going to produce these spindle fibers, which I will represent with these yellow lines. And they're going to move toward the opposite poles of the cell. So this is why I actually started drawing them onto different sides of the cell. So they would actually be floating in the cytoplasm of the cell, and they would come down to these opposite sides. And now the nuclear envelope is going to start breaking down. So remember, your DNA was inside the nucleus. Now the nucleus is starting to break down and exposing these chromosomes. So now moving on to prometaphase, these chromosomes are going to continue to condense. So I'll draw those chromosomes. The kinetochores are going to appear at centromeres. So centromeres, they are at the middle of each of these chromosomes. And the kinetochores are going to appear here. So this is where the spindle fibers are going to connect to each of the chromosomes. So kinetochores form. I'll just write that down. Kinetochores. And then the mitotic spindle is going to attach to the kinetochore. So again, centrosomes. And then our spindle fibers, they're going to start to connect to these. So that is prometaphase. And then in metaphase, these chromosomes are going to line up at the metaphase place. So metaphase is essentially, meta means middle. So these chromosomes are going to line up directly in the middle of the cell. Don't forget, our spindle fibers are still connected to each of these chromosomes with the kinetochores. And each sister chromatid is attached to a spindle fiber from opposite poles. So sister chromatids, this is basically referring to the chromosome I have here. So let's say that I blew up this image of the chromosome. We would have, it would kind of look like this. This is the chromosome. Sorry. And then this would be two sister chromatids. So each of these is going to essentially be connected to separate spindle fibers. And so in anaphase, this is when they are going to be pulled apart. So the centromeres split into two. The sister chromatids are now pulled toward the opposite poles. So remember our centrosomes. They're going to start to pull, or they're going to pull these chromatids apart. And then lastly, in telophase, the chromosomes are going to arrive at opposite poles, be condensed. The nuclear envelope is going to develop around each of the set of chromosomes. The mitotic spindle is going to break down, and the spindle fibers are going to continue to push the poles apart. So what this looks like We have these. We have our spindle fibers, and then we have these chromosomes. And the nuclear envelope is going to start to form around them. And this is essentially going to form separate nucleuses for each of these daughter cells. So in order for these daughter cells to form, cytokinesis needs to happen. So what's happening with cytokinesis is it's going to break these two cells apart. It's going to cleave them right in the middle. And then we will, in the end, have two daughter cells. And these will each have their own copies of DNA. And so when we are growing from infants to adults, obviously we're not the same size as we were when we were infants. A lot of cells had to be produced in order for us to grow. So in order for these cells to be produced, mitosis had to happen over and over and over and over again, producing more and more daughter cells and eventually building up our body. So included in the module for module 6, there was an interactive that was provided, and this is with Howard Hughes Medical Institute. And I'm actually going to share this interactive if you haven't already checked it out. So essentially, this is showing you the different phases of the cell cycle. So of course, we have our interphase, which I said before includes the G1 phase, the S phase, and the G2 phase. And then we have the M phase where mitosis is going to occur. And then something that you should know is how mutations can occur and how they can affect this cell division. So there are two different types of proteins that are covered in this interactive that are discussed. So we have our stimulating proteins, which are proto-oncogenes, and our inhibitory proteins, which are encoded by tumor suppressor genes. So I would definitely check this out. I would definitely know, I would definitely go through and know about these different proteins and know how they can cause cell division to, essentially it won't work properly and your cells will divide and divide and divide and they can eventually form tumors, which is very unfortunate. And this is cancer. Okay, so I'm going to go back to my other screen. And remember, stimulating proteins, they are encoded by proto-oncogenes. And then inhibitory proteins, they're encoded by tumor suppressor genes. And that's pretty much it for Module 6. Do you guys have any questions before I move forward into Module 7? Okay, so I'll move forward. And just a reminder, if you have any questions, you can feel, you won't really have to know any specific ones. I would just know stimulating proteins are encoded by proto-oncogenes and inhibitory proteins are encoded by tumor suppressor genes. Yeah, you don't really have to know much in detail. Yes, and genes are small parts in the chromosome. They're the nucleotide sequences that are within the chromosome. Okay, are there any other questions? Okay. So I would definitely just go through this interactive that I showed earlier, and I would just know, it'll tell you how these genes are mutated and how they can cause cancer. So yeah, I definitely read into it on there. Awesome. Okay, any more questions before we move forward? Great, thank you guys for asking. And don't be afraid to ask questions. I will be checking the chat, like I said, periodically, and I will answer them as soon as I can. Okay, so moving on to Module 7. This module basically covers sexual reproduction. We're going to be talking about meiosis. So meiosis and mitosis, they sound similar, but we'll be going over the differences and the similarities. We'll also talk about genotypes and phenotypes. So we'll go into Mendelian laws and then also into non-Mendelian inheritance patterns. So starting out with sexual reproduction, why would animals want to use sexual reproduction? Essentially, sexual reproduction is going to provide offspring with genetic diversity. So with binary fission in prokaryotes, these are not going to undergo sexual reproduction. They're going through asexual reproduction, and they're just creating identical copies of each other. Whereas with sexual reproduction, each of the offspring from the parent, they're going to be different genetically from them, which can help because let's say that there was an offspring and there was a certain sickness or disease that came around, the offspring that has a mutation or even just because it has different genes, there's a higher chance that it's going to survive from that disease than the offspring that definitely would not survive that all had the same exact genes. So what happens during sexual reproduction is the production of gametes. And so these gametes, they are produced by meiosis. And gametes are essentially your egg and your sperm. So meiosis, it's a little different than mitosis in the sense that you're going to be creating these cells that only have half of the amount of chromosomes than a parent cell. So we as humans, we have, like I said before, 46 chromosomes. We are diploid organisms, but when we have our offspring, the gametes, they're going to be haploid. They're going to be 1N, as referred in the module. So when you have two parents, you have the male and the female come together, they are going to create that diploid cell. So the process of meiosis, the goal of meiosis is to take the parent cell and break it down into these haploid cells. So in order for this to happen, we need to have two stages of meiosis. We have meiosis 1 and meiosis 2. And the reason these, I say, are similar to mitosis are they go through the same phases. We have prophase, metaphase, anaphase, telophase, and cytokinesis, all going on in each of the steps of meiosis. So the difference between meiosis and mitosis is you're going to notice that the phases for meiosis, they're going to either be specified as prophase 1 or prophase 2. So I'm just going to write these in over here. In this case, in meiosis 2, you're going to have prophase 2, metaphase 2, so on and so forth. And one of the biggest things that happens in meiosis is crossing over. So this is where you're going to have that genetic diversity happen. And this is the reason why your parent cells are going to be so different from the offspring cells in sexual reproduction. And this is because these chromosomes, they're going to cross over and basically randomize the genes, and thus the offspring is going to have randomized genes from both the female and male parent. So I'm going to go through meiosis 1 step by step, and essentially the steps in this process are very similar to mitosis. So I'm actually going to, okay, awesome. So in meiosis 1, we're going to start with one parent cell, and the parent cell is going to have the paternal chromosome as well as the maternal chromosome. So the paternal chromosome is going to come from the male parent, and the maternal chromosome is going to come from the female parent. And so I'm going to represent these with the colors blue and pink, just so that we can specify which ones are which. So the paternal chromosome here, the maternal chromosome is here. And so what's going to happen in meiosis 1 in prophase is these cells are going to cross over. So what's going to happen is the female chromosome, and the male chromosome, they're going to randomly cross over, giving each other random genes. So if I were to redraw these chromosomes after crossing over, this would now have parts of the paternal gene incorporated into the maternal chromosome. And the same goes for the paternal chromosome. So I know it's a little bit hard to see, but basically this is why we are so genetically different from our parents, because each of their chromosomes from their mother and their father, they both crossed over. They have completely different genetic makeup compared to ours. So now in meiosis, we're going to go through the process of our prophase, metaphase, anaphase, telophase, just like in mitosis, essentially. But now we have these genes and these chromosomes that are completely different than the parent. So we are going to have, so we have prophase when the chromosomes are going to form. We have our mitotic spindle forming as well. We have metaphase, or actually, sorry, I drew too little of these. This would be prophase. We have our chromosomes condensing, and now imagine that each of these chromosomes has crossed over. And the mitotic spindle is going to form. And then we have metaphase, where these chromosomes are going to line up in the metaphase plate in the middle of the cell. And now our kinetochores are going to form, and the spindle fibers are going to connect to each of the centromeres and the kinetochores in the middle of those chromosomes. And then we have anaphase, where it's going to start pulling these chromosomes apart. And actually, I'm sorry. Just to make this clear, we're going to keep our maternal and our paternal chromosome. So here's our paternal chromosome, and here's our maternal chromosome. Remember, they have undergone crossing over. They're going to line up in the middle of the plate, and the spindle fibers are going to connect to the kinetochores, and then they are going to be pulled apart in anaphase. And just imagine that these were crossed over, just to keep things easy. Okay. So now after our anaphase, they're pulled apart, and in telophase, that nuclear envelope is going to form, and now we have two cells essentially with one of the paternal chromatids and one of the maternal chromatids. Because remember, the sister chromatids are being pulled apart. So that is meiosis 1. The result of meiosis 1 is we have two cells, and each of them is going to have one of the maternal chromatids, one of the paternal chromatids. But in order to form gametes, we need them to be haploid. So these are still diploid because they have both the maternal and the paternal chromatids. So meiosis 2 is going to occur with these cells. And in the end, the result is going to form 4 haploid cells. So they're only going to have one set of chromosomes. Each of these is going to have 23 chromosomes in total. And these are the maternal and the paternal is just representing exactly 23 chromosomes, because remember, you get 23 from the maternal side and 23 from the paternal side. So in total, meiosis forms 4 haploid cells. And they are all genetically different from one another. So that is the process of meiosis. I know meiosis is a little bit difficult to explain. Does anyone have any questions before I go forward? There is prometaphase, but we don't really talk about it because it's just kind of the in-between between prophase and metaphase. So it's just a little bit more specific. So it would be the same as the process of mitosis in the sense that prometaphase is when the chromosomes are going to condense and the kinetochores are going to appear. So it does happen, but we're just saying, like, we're just going through the process in general and just going prophase, metaphase, anaphase, telophase. But prometaphase is still happening in between. Okay, any other questions? I hope that made sense. Okay, we will move forward then. So now we're going to go into Mendelian genetics. We're going to talk about genotypes and phenotypes. So genotypes, the genotypes is what's in your DNA, what's in your genes, and these are going to be represented by alleles. So you can see here, I drew 2 alleles, and these are both represented by capital letters. This is RR, and so we're going to talk about the color of a flower. So in this case, R is going to represent red, and you can notice that both of these R's are capitalized. So that indicates that these are both dominant. And because we have 2 alleles that are the same, they're going to be homozygous. So these alleles are going to be homozygous dominant. In the next genotype that's listed, we see that there's a big R, which represents the dominant allele. We also see a little r that is in a gray color. I wanted to represent a white flower, but because it's a white background, I had to use gray. So let's imagine that the little r represents white. So this is an uncapitalized R because it represents a recessive allele. So because this gene in the middle has alleles that are dominant and recessive together, this is going to be referred to as heterozygous. Where hetero means different and homo means the same. So in the last set of alleles, we see that there are two of the same alleles in the capitalization. So they are both the same, they are homozygous, but since they are little r's, they are recessive. So these are homozygous recessive alleles. And these genotypes, like I said before, your genes can essentially determine what type of traits that you possess, and the traits that are expressed are what we see. They can also be referred to as phenotypes. So with these flowers, in Mendelian genetics specifically, we have our homozygous dominant showing that we have a red flower. And then in our heterozygous dominant, we also actually see a red flower because the dominant gene overpowers the recessive gene, and the trait that is expressed is dominant. So actually, when Mendel was doing his pea plant experiments, which were covered in the module and which we'll go over briefly, he expected that the offspring were going to be a mix of each of the parents. So let's say that we have like the green pea versus the yellow pea, and he expected the offspring would be an in-between between the green and the yellow. But what he found was that the green pea, which had the dominant alleles, was expressed in both of the green peas that had completely dominant homozygous alleles and also in the heterozygous alleles. And that's because the dominant gene takes over and expresses that trait. Whereas with the homozygous recessive gene, now there's no dominant gene that can possibly be expressed. So the resulting phenotype, what we would see in a flower in this case that has two homozygous recessive alleles for color, this is going to be a white flower. So that's essentially the difference between genotypes and phenotypes. So Mendel, like I said before, he did his pea plant experiments, and I'll actually pull up a little picture to better explain it. Sorry about that. Hold on one second. Okay, so this is just showing Mendel's law of segregation, which you may have seen on the last slide that I showed. And essentially you can see here, we have in this case, the green pea and the yellow pea. Actually, I had it mixed up, sorry about that. The yellow pea is dominant and the green pea is recessive. So when the green pea and the yellow pea are crossed, or parents reproduce, they create an offspring that is going to usually be have a phenotype of the yellow pea because the yellow is dominant. And with two peas that are recessive, you're going to see that the resulting pea offspring is going to express a recessive trait that is green, and so on and so forth. And so essentially, Mendel's law of segregation, which I'm going to share another picture. Only one of the two alleles for a gene is put into a gamete, and at fertilization, offspring receive from each parent one allele for each gene. So going back to our other presentation, sorry. So the definition for our law of segregation is during gamete formation, the alleles segregate from each other, and each gamete has an equal probability of containing either allele. And for this, I would just know this definition. And if you need to refer back to it, I'll post the video. And then for Mendel's law of independent assortment, it's pretty much the same case. Every possible combination of alleles for every gene is equally likely to occur. So these are Mendel's law of segregation and Mendel's law of independent assortment. Each allele is equally likely to occur. And this is the case that he saw in his pea plants, like we said before. Each allele, you could either have dominant or recessive. It's equally likely to occur in each of the gametes. And then going into non-Mendelian inheritance, this is when things are a little bit different than what Mendel saw in his pea plants. And so some genes, they can be inherited in this non-Mendelian way, and they can be expressed a little bit differently than at least Mendel expected. So going into incomplete dominance, here we have our flowers again. We have our red flower, which, like we mentioned before, is our homozygous dominant, and then we have our white flower, which is our homozygous recessive. So when we mix these two together, which I'm going to represent using a Punnett square, we will end up with a heterozygous allele for each of the resulting plants. So what you would expect is for all of these to be expressing a red flower, right? Because the dominant gene should take over. But with an incomplete dominance, this is actually not the case. Instead of the dominant allele being expressed in the phenotype, the recessive allele is also expressed at the same time. So the resulting flower color would actually be pink. So it's the intermediate between the dominant and recessive allele being expressed. So that is incomplete dominance. Moving forward, there's also codominance. This is another non-Mendelian inheritance, sorry. So with codominance, let's say that we have two different cows. So these cows at the top, these are the parent cows. We have a black cow, completely black coat, and we have a white cow with a completely white coat of fur. And let's say that these cows come together and they create offspring. Using a Punnett square, we would put together each of these alleles, and we would see that each of the cows or each of the offspring, the chances of it having like certain alleles, it would have both alleles for black and white. But with codominance, it's not going to be like Mendelian genetics. It's not going to have one gene dominating the other in the phenotype. In codominance, it's going to express both of the genes. So in this case, the resulting offspring would express both white fur, essentially, and black spots. And so this is an example of codominance. And this is also something that you would see in certain cats. So calico cats, I believe, also express codominance. So they express orange and white, if I remember correctly. But yeah, that is codominance. All right, and then going into multiple alleles. So a good example of multiple alleles would be with blood type. So in blood type, there are actually 3 different alleles that can determine your blood type. We have A and B, and we have O. And so A and B alleles, they can be codominant, and your O is going to be recessive. So the blood types that are resulting from multiple alleles, you could have blood type A, you could have blood type B, blood type AB, or blood type O, so on and so forth. And so this is because we have multiple alleles working. And then lastly, yes, we're going to talk about sex-linked genes. So with sex-linked genes, these are genes that are going to be on the sex chromosomes. So a good example of this would be with your red-green color blindness or hemophilia. And hemophilia is basically when the blood does not clot correctly. So if you have a cut, you could bleed excessively because you're never going to form a scab over that cut. So these genes are sex-linked. And so that means that usually they are on the X chromosome. So here we have a regular female set of chromosomes, sex chromosomes, which are XX in a human. And then in males, we have our XY chromosome. And so when an X chromosome carries a disease such as red-green color blindness or hemophilia, it'll be on one of the X chromosomes. And so I have that represented here at the bottom. And we will represent this X by XC, and the C is in this case going to just stand for carrier. So if this X chromosome, let's say that this female right here had offspring with this male right here, the offspring, there's a 50% chance that it's going to be male, 50% chance that it's going to be female. But because one of the X chromosomes on the female had the carrier gene, there's also a 50% chance that the female being produced would be a carrier. And there's also a 50% chance that the male offspring would have the disease. So the reason why males, like if let's say this female, we call her a carrier and not necessarily infected or having red-green color blindness or hemophilia in this case, they wouldn't have this because they have this other X chromosome working. And so that chromosome doesn't have red-green color blindness, doesn't have hemophilia, and that can be expressed instead so that the female does not have either of those diseases. Whereas the male, they only have one X chromosome to get their information from. So if this male had the carrier gene, it would have to express it. And thus, that's why more men are usually prone to having red-green color blindness compared to females. And that is sex-linked inheritance. So that was pretty much covering all of Module 7. Are there any questions before I move forward? Okay. And if you're going to go back and look through all of these, I'd definitely just briefly go through the videos and just cover each of the topics that we went over with Mendelian genetics, non-Mendelian inheritance, and you should be fine with those. All right. So before we move on to Module 8, I know it's been about an hour since we started, so I will let you guys have a break. We'll take a 10-minute break, so you can come back at 5:08. And in the meantime, I will actually answer this question in the chat. So genotype is the trait gene-wise, and phenotype is the trait in physical form. Exactly, yes. So the genotype would be represented by the alleles for that trait, and the phenotype is the trait that you would physically see being expressed. Exactly. Okay, so if there's no further questions, I will see you guys back at 5:08. Thank you. Oh, and I actually forgot to record. So I will resume the recording from here, but this is just the digestive system, and this is the whole process going through step by step, where the digestive tract starts and where it ends. And in the last slide, we talked about homeostasis, which is just your body balancing everything. All right, so this is the digestive system. So moving forward, we're going to talk about the respiratory system. So the respiratory system, this is going to include your lungs, your pharynx, your nasal cavity, your trachea, and this is where you're going to bring in oxygen to your body because your cells need oxygen in order to function, and you are going to release carbon dioxide. So carbon dioxide in our cells is a waste product, and so our body needs to release it. So with the respiratory system, I would definitely know the different parts that the air or gases go through. So when you breathe in, the air is going to first come into the nasal cavity. And then the air is going to move down into your pharynx, which is kind of right at the beginning of where your throat starts. And then it's going to move down into the trachea, which eventually goes down into the lungs. And within your lungs, you're going to have these branches coming off. You're going to have your bronchioles, and there's going to be gas exchange. So your blood is actually going to travel through these capillaries that are within your lungs, and they're going to take in the oxygen that you breathe, and they're going to release the carbon dioxide. So when you breathe out, you're going to release that carbon dioxide from your body. So this is the respiratory system, and I would know these parts. Moving on to the circulatory system, I'm so sorry in advance. My heart drawing is a little rough, but I hope we can make sense of it. So this is the heart, and this is kind of the main part of the circulatory system, which is why I drew it. So your heart is going to take blood, which I mentioned before, from the lungs. It's going to take that oxygen, which it needs to give to cells, and then it's going to release that carbon dioxide. So the blood that is oxygenated, which means it has oxygen, it's going to distribute to the rest of your body. It's going to give all of your cells oxygen, and then each of the cells is going to give the blood whatever waste products it has, which is carbon dioxide. And that means that the blood would not be oxygenated. So here in this diagram, you're going to notice that there's a red part of the heart and there's a blue part of the heart. And so the red part of the heart actually represents the blood that is oxygenated, and the blue part represents the blood that is deoxygenated. And so the amount of oxygen that is within your blood can actually determine how red that blood is. So looking at this heart, I also wanted to specify that the part where there is deoxygenated blood, which is blue, is on the right side, and the part which is red with the oxygenated blood is on your left side. So looking at this, you might be like, oh, well, the red side, it's on my right side. You have to think of it as if the part that you're looking at is, let's say, like in the person standing right in front of you and they're facing towards you, their left side would be opposite to your left side in that case. So the left side is where the blood that is oxygenated is going to be pumped into the rest of the body, and the right side is where the blood that is now deoxygenated, has that carbon dioxide, is received from the body. And so going into the parts of the heart, we have 4 different chambers. So I numbered them 1, 2, 3, 4. And out of these 4 chambers, 2 of them are the atria or atrium, 2 atrium. And we have 2 ventricles. So blood is going to come into the atrium and it's going to be pumped out of the ventricles. And so actually, I'm going to pull up a diagram of the heart that's a little bit clearer than the one that I drew. I tried my best, but it's a little bit, I want to make sure that it's really clear to see. So this is a picture of the human heart. This is a lot clearer than the one that I drew. And you can see there's the right atrium, there's the left atrium, there's the right ventricle, the left ventricle. You can also see that the left side has that oxygenated blood. The right side has the deoxygenated blood. And like I said, the blood is going to enter the heart through the atrium, and it's going to leave the heart through the ventricles. So in the case of the right ventricle, it's going to go to the lungs, and then the lungs are going to oxygenate the blood, and that blood is going to come back to the left atrium. And then from the left atrium, it's going to go to the left ventricle, and the left ventricle is going to pump that blood out to the entire body, giving all of those cells oxygen. So that is the circulatory system. So let me go back to sharing my other screen. Are there any questions before we move forward? Okay. So going forward, we're going to talk a little bit about asexual versus sexual reproduction. So these were mentioned before, sexual reproduction, especially in detail. And remember, sexual reproduction, the offspring that's going to be created is very genetically diverse. These offspring are going to be way different from the parents genetically, and they're going to have a higher chance of survival because of it. Whereas with asexual reproduction, in the case of like bacteria or even plants, they're going to produce identical copies of themselves. So these are going to be identical, genetically identical offspring produced from asexual reproduction. Next, asexual reproduction, each individual cell can produce its own offspring, or each individual, maybe in the case of a plant, each individual plant can produce its own offspring. And in bacteria, each bacteria cell produces its own offspring. But in sexual reproduction, only the female is able to produce offspring because the female has the reproductive organs that allow for it to produce the offspring. And then next, going into the amount of offspring that asexual versus sexual reproduction can produce. Asexual reproduction can produce a large number of offspring. So if you think about bacteria growing on a petri dish, let's say that you swabbed, a few bacteria colonies on a petri dish and you just left it there for a few days, there are thousands and thousands and thousands of colonies that are produced within even the span of a few hours. Whereas in sexual reproduction, it takes humans, in the human's case, 9 months to produce a child. So it takes lots of energy to produce offspring. So there's few offspring that can be made in comparison to asexual reproduction in one span of time. So those are the main differences between asexual and sexual reproduction. And of course, they're similar in the fact that offspring are being produced and genes are passed down. So this module also briefly talks about the types of asexual reproduction. So there are 4 different types that we talk about. First off, we have fission. And in the lecture video that was posted for this, the example was with sea anemones. So basically, this is when the parent would split into 2 parts and regenerate. And like we talked about with bacteria, with binary fission, remember the bacteria pretty much just splits into 2 and it then creates 2 different bacteria cells. And then we have budding. So this is when a bud of the organism from the original organism forms, and the bud is much smaller, and it eventually grows into an adult, into a new adult. Then we have fragmentation. And the example provided was with starfish, so I drew a little Patrick star. And basically, when these animals are broken into parts, they can regenerate. So let's say that a piece of Patrick was cut off, maybe by a predator, or I don't know, he fell on something. Good old Patrick. This piece could regenerate into a completely new starfish. So that's fragmentation. And then lastly, we have parthenogenesis. And this is done by bees. So in this case, the egg does not need to be fertilized. And so in the case of bees, I believe if the egg is not fertilized, it becomes a male bee. So those are the types of asexual reproduction, and a resource that is really good to go to if you would like to cover these in a really fun way is on the Google Docs that I posted under extra exam resources. There's a game that I found that you have to go through. You have to go through a form of asexual reproduction and you have to complete a puzzle, which is really fun. So if you're interested, I would refer to that document and play the game. Super cool. All right, so now going into sexual determination. So we've kind of briefly mentioned this before. So humans, when there's a female human, they're going to have XX chromosomes, sex chromosomes. And for the male human, they're going to have an XY chromosome. And so this is actually not the case for all animals in the animal kingdom that go through sexual reproduction and have sexual chromosomes. So an example I'd like to bring up is with birds. With birds, instead of having XX and XY, they have ZW and ZZ. So if they have a ZW, they are female, and if they have a ZZ, they are male. And then you may be thinking, what about people that are intersex? So humans that are intersex, they have, in this case, they could have 2 X chromosomes and 1 Y chromosome. So some traits that are expressed in people that identify as intersex, they have genitals that don't fit male or female category, so they may not have the same types of genitals by looking on the outside, or in some cases they may have either male or female looking genitals, but internally their organs don't fit the male or female category. So now moving forward with fertilization. Fertilization can happen outside of the body, which is referred to as external fertilization, or fertilization can happen inside the body. So with oviparity, this is actually when animals develop outside of the body. So a good example of this would be with sea turtles, right? They lay their eggs and the turtles develop within those eggs outside of the body. And then with ovoviparity, I hope I'm pronouncing that correctly, that's essentially when the fertilized eggs remain in the female until they are hatched, or these eggs are laid right before they hatch. So this is in the case of some sharks. Some sharks will actually keep their offspring in their eggs right before they're about to hatch from the eggs. They lay the eggs. And then with viviparity, this is basically live birth. So this is what humans undergo, squirrels also undergo, and this is live birth. Gametes are used to form zygotes, and this is when 1 sperm and 1 egg come together. So these are the different types of fertilization. And then briefly, I just wanted to go over gamete production. So again, in females, the gametes that are produced are eggs, and in males, the gametes that are produced are sperm. And in females, these would be produced in the ovaries, and in males, these would be produced in the testes. And when the egg and sperm come together and they are fertilized, they form a zygote. So I actually took one of the pictures straight off of the videos that was given for this module, and it's just showing the stages that the zygote goes through. So the zygote starts right when the fertilization occurs, and then the cells are going to undergo mitosis. They're going to divide, and eventually they are going to create a blastopore and a gastrula. So the gastrula, these are going to be cells that are going to differentiate into different organs by organogenesis. So when you are developing your cells, like when they're first fertilized, they don't know exactly what they're going to be. They could be neurons, so like brain cells. They could be skin cells. They could be cells that are with your heart, your cardiac cells. So before they can differentiate, they need to become a gastrula, and they need to then differentiate by organogenesis. And then the lecture video also talks about the male endocrine system, and the female endocrine system. So I just wanted to go over the different hormones that are involved in these processes. So a really great example and diagram is included within that video. So this is kind of where I pulled it from. But essentially, these are the male reproductive parts. This is the penis, the testes, and then there's the brain and the hypothalamus. So this is the endocrine system. This is going to be giving hormones to these different parts of the body when needed. So first off in the process, your hypothalamus, this is going to release your GnRH, which is gonadotropin releasing hormone. And when this gonadotropin releasing hormone reaches the anterior pituitary gland, the pituitary gland is going to release these two hormones called FSH and LH. So these are going to stimulate sperm production in the testes. And then let's say that the amount of sperm, like your body has created, or at least the male body has created enough sperm that it needs. There needs to be some kind of regulation for it to stop. So then the testes are going to send a signal to the brain saying, hey, let's stop this. Got to stop sending this FSH and this LH down here so we can stop producing too much sperm. So the testes will send these hormones referred to as inhibin and testosterone to the hypothalamus, to the anterior pituitary gland, and they'll be like, we need to stop sperm production. And so then the body will stop creating sperm. And then let's say that, again, the sperm needs to be produced and there's a low level of it. What the body would do is it would stop sending these inhibin and testosterone hormones to the hypothalamus and anterior pituitary gland. Those levels of those two hormones is going to go down. And then the hypothalamus will now be stimulated to, again, release GnRH, and then stimulate the anterior pituitary to release LH and FSH, and the whole process will start all over again. So that is the male endocrine system. And then we have the female endocrine system. So in the female endocrine system, we have different phases actually. So of course your hypothalamus is going to release the gonadotropin releasing hormone, and the anterior pituitary gland is going to release FSH and LH to the ovaries. And here, so in the beginning of a woman's menstrual cycle, this very beginning is called the follicular phase. So a female is going to produce only one egg per month, whereas males are going to produce thousands or even millions of sperm per even day. So this happens in a very long cycle. So in this first phase, which is referred to as the follicular phase, the LH and the FSH are going to go to the ovaries, and they are going to stimulate follicles to grow. That is why it's called the follicular phase. And then next, the body is going to go into the ovulation phase. So the LH and the FSH in the ovulation phase, they're going to stimulate the maturation of one of the growing follicles. And so then from the ovaries, they're going to send estradiol to the hypothalamus in this phase. And they're also going to send it to the uterus to create that lining. So in ovulation, the egg is actually going to be released from the follicle, and the uterus is now going to be thickened. It's basically going to prepare for offspring to be created. And then lastly, in the luteal phase, this is when the LH stimulates growth of the corpus luteum, forms follicular tissue, left behind after ovulation. And there's going to be instead of a positive response with the estradiol, actually, this is the luteal phase. The ovaries are going to tell the brain to stop releasing the gonadotropin releasing hormone, stop producing estradiol, progesterone, and the corpus luteum is going to break down. It's going to block the gonadotropin releasing hormone, and the endometrium will further develop, and eventually it will be shedded away. And this is when the menstrual cycle releases the wall that's within the uterus. So that is a very brief recap of the female endocrine system. I would refer back to the video because it's very helpful. And I would just know, I'd be familiar mostly with the specific hormones that are used in each of these processes, and then also knowing about the follicle that's in the female endocrine system. Okay, are there any questions before we go on to chapter 9, or module 9, sorry? Okay, I'm gonna keep going with module 9. As I said before, module 9 is going to be offered with the exam. I would highly recommend to go through the entire module before taking the exam. There's going to be quite a few questions that are referring to module 9, so it's really important to be familiar with the content that is in that module before you take it. So yes, go through the module, and I'm going to go through it pretty briefly. I don't want to go into detail just because you're going to have to go through the rest anyways during the week. So this module is basically talking about viruses. So viruses, they're microscopic particles that infect, hijacks, and replicates inside of a host cell. So the two main parts of the virus, there's the protein coat, which is also referred to as the capsid. And within that protein coat, there is genetic material. And there are many different types of viruses, different virus shapes as well. So this is a chart with virus morphology. In the video that will be offered in the module, it is going to go into detail with all of these, but these are all of the different shapes. So we have helical, polyhedral, spherical, complex, which is also a bacteriophage. And also listed in this chart are the different types of viruses that take on these specific shapes. And so, as we know, COVID-19 is really big right now. We are going through a pandemic. And so COVID-19 virus, it is actually a spherical virus. And then briefly covering COVID-19, there will be a question about this on the exam. COVID-19, also on top of it being a spherical shaped virus, it is speculated that it was transferred from bats to humans. And COVID-19 also infects the lungs. It specifically targets the ACE2 receptor, and this receptor affects blood pressure. Okay, so moving forward with viral replication, these are basically the steps that are taken by, in this case, bacteriophages to replicate. So the bacteriophage is going to attach to the surface of the host cell. It is going to penetrate the cell, and it's going to inject its DNA within that cell. And then it is going to hijack the cell system, and it's going to replicate its own DNA and create more of its proteins, creating more bacteriophages. And eventually these bacteriophages are going to mature, and eventually they are going to cause the cell to burst, or in this case, it's referred to as lysis. where the cell lyses. So this is viral replication of a virus that only occurs in bacteria. That is why I say bacteriophage. So you wouldn't find this happening to eukaryotic cells. But let's say that there's a bacteria inside your body, a virus could come to that bacterial cell and it could reproduce this way. Okay, and then basically one way that we can stop viruses from infecting bodies or certain organs is by using vaccines. So vaccines are substances that stimulate the production of antibodies and provide immunity against one or more diseases. So they can help our immune system by providing it with certain antibodies. These antibodies give information to the body of how to combat these viruses. And in the video, it's going to go through a few different types of vaccines. So I would just briefly know about those, but overall, I would just know what a vaccine does. And then the module also talks about innate versus adaptive immunity. So in innate immunity, innate means something that you're born with. So these are not specific. These will target viruses that are not specific. They could target things that are not caused by infection. And they mainly depend on physical and chemical barriers in the body. And then with adaptive immunity, this is what you're not born with. This is in the case of your body learning how to fight off a specific pathogen or a sickness. So it can target a specific pathogen that can be caused by infection. It does take a little bit longer to respond in this case, but with adaptive immunity, your body will have a memory system that will learn how to combat that pathogen. Okay. All right. Last few slides. So when a pathogen is recognized by the body, chemicals called cytokines sorry, the app that I used for the other one only went up to 50 slides, so I have to continue on notes. Cytokines are produced to tell other immune cells to fight off infections. So I would know about these cytokines. These are going to be mentioned in the lecture video provided. Also know about APCs, sorry. APCs are antigen presenting cells, and these are going to detect, engulf, and inform the adaptive immune system if there are different pathogens in the body. And then lastly, I would know about B cells and T cells. I believe you're going to have a project that deals with these two, but to give an overall definition, B cells target infections in blood and lymphocytes. Or blood and lymph. They have a humoral response. And then T cells, they target cells as part of the cell-mediated immune response. So that is a very general overview of the definition for these two. These will go, you'll go into a lot more detail with the video that's provided. But overall, I would say that that's pretty much everything you would need to know for the exam. I've covered all of the main topics that are on there. It is 45 questions long. I don't believe there's a time limit, or if there is a time limit, it would probably be around maybe an hour or so. But from this point on, I would definitely watch the lecture vid on how your body fights disease. This will be in Module 9, which will be released on Friday. And also definitely complete Module 9 before the exam. So if you guys have any other questions, feel free to ask. But otherwise, thank you so much for coming. It means so much. And I hope you guys come again. And if you have any other questions throughout the week, you can feel free to come through my office hours. My information will be on Blackboard. But thank you, and I hope you have a great rest of your week. Thank you. Thank you, guys.